Hello class 9. We will resume our study of the seed chapter from the germination part. Okay. Now I hope you all know what is germination. It is actually the formation of a seedling developed from the embryo, and it is only possible when it is getting proper conditions, favorable conditions. The seeds contains inside it a dormant embryo, that means an inactive embryo. In a dry seed, the embryo is inactive. It is said to be in a state of dormancy or period of rest. It appears to be without life. The dry seeds consume oxygen, gives out CO2, release some heat as well. But when they are placed in proper conditions, it becomes active and starts growing into a seedling. Seedling means when from a seed a very tiny plant comes out, and all the changes leading to the formation of a seedling, we collectively term it as germination. Now, for germination, some of the conditions which are necessary, we must know that. First one, we all have this idea that water is the most important natural condition, which is required for germination. And the two main uses of water are the seed swells and consequently the seed coat ruptures, allowing the elongating radicle to come out and form the root system. Water is necessary for chemical reaction and for the enzymes to act upon the food stored in the cotyledons or endosperm so that it may convert into a diffusible form dissolved and utilized by the growing embryo. So many chemical reactions occurs inside the seed while it is growing into a seedling. So for that, water is very necessary, and a lot of enzymes are reacting in that. Those enzymes are made up with water and they act upon the food stored in the cotyledons with this water. Next is suitable temperature, because very high or very low temperatures both are unsuitable for germination. So we need an optimum temperature, a moderate temperature, which is 25 to 35 degree centigrade. Another one which is very important matter, which is oxygen. It's an element which is without which we cannot think of even germination. And during germination, it's the process of rapid cell division, rapid cell growth, for which energy is required. So this energy is available only by respiration or oxidation of the food, and hence the need for oxygen. Clear? Now, we have to go through various experiments. Now let us see one diagram. First, I am starting with this experiment, which is a 3 bean seeds experiment. Why I am starting with this? Because it's a very important experiment for your exams. So I am starting with this experiment first. See, here we have taken a beaker and 3 mature air-dried bean seeds we have taken and tied to a glass slide in 3 positions. Now this is kept in a beaker that is containing water in a manner that the top seed is above the water, middle one is just at the water level, and the bottom one is deep inside the water. Now what we will observe after a few days, the middle seed germinates as it gets both oxygen and water. The top seed does not germinate at all because it is getting only oxygen but no water, which is a very necessary factor. And bottom seed does not germinate or stops germinating. It gets water but very little oxygen, without which it is not possible to germinate. So this experiment conclusively proves that water is essential for germination, but the other requirement of oxygen is not fully demonstrated. So in this experiment, actually we are trying to prove that when a seed is getting both oxygen and water properly, then only it can germinate well. But if it is getting any one of them, then it will not do so. Clear? So it's a very important experiment, and please, this diagram is very important. You have to practice this diagram. Now I'm coming to the first experiment. Here we have to prove that water is necessary for germination. We will take two beakers and we'll mark them A and B. Okay. In beaker A, we'll place some seeds on wet cotton wool. In beaker B, again we'll place some similar seeds on dry cotton wool. Keep both the beakers in an ordinary room. In a day or two, we'll see the seeds in beaker A will germinate and not in beaker B, showing that water is necessary for germination. That means what? Wet means that is supplying continuous water, and dry means that is not supplying water. So that means water is necessary for germination. That is why the one which was kept on the wet cotton wool, that started germination. But the beaker B seeds does not show any germination. Okay, this is a very simple experiment to prove water is necessary for germination. Next experiment to prove that suitable temperature is necessary for germination. Again, we are taking two beakers, naming them A and B, placing some green gram seeds on wet cotton wool in each of the two beakers. Keep beaker A in an ordinary room and beaker B in a refrigerator. In a day or two, what we'll see? The seeds in beaker A will germinate, showing the importance of a suitable temperature for germination. And the seeds in beaker B may not show signs of germination or may germinate after several days, though not to the extent the seeds in beaker A germinate, because beaker A will germinate properly. Why? Because it is getting an optimum temperature, not very less. But beaker B, the seeds are getting a very low temperature, which is not suitable for germination. So that is why it will not germinate properly. Clear? Next experiment to prove that oxygen is necessary for germination. We are taking two conical flasks, you see the diagram, and naming them A and B. Spread wet cotton wool in each flask and place on it some soaked gram seeds. Lower a small test tube containing alkaline pyrogallic acid. Now what is the use of this acid? It is an organic acid. It absorbs oxygen. And in flask B, taking care that not a single drop of the chemical falls on the seeds or the cotton wool by means of thread. Okay. Keep the tube hanging by fixing a cork on the mouth of the flask. Arrange flask A in the same way, except that the test tube in this flask contains plain water. Place the two flasks in an ordinary room. The seeds in flask A will germinate, showing the importance of oxygen for germination. Because in flask A, we have kept the plain water test tube. And in flask B, we have kept the test tube with alkaline pyrogallic acid, which has absorbed all the oxygen. Now, the seeds in flask A will germinate because their oxygen is present, and the seeds in flask B do not germinate because there is no oxygen. So, this proves that oxygen is necessary for germination. So, we are done with all the experiments today. Okay. Now students, in the next class, we'll go to the types of germination, their diagrams, and we'll finish off the chapter next day. So today up to this, and please go through the experiments properly. If any problem come up with that problem to me, I'll definitely solve it. Thank you.